So, okay, class, can we continue? Boleh continue tak?
Yes.
Boleh ya?
Yes.
Kita pergi ke gene expression, transcription. Okay? So, what is transcription? Apa yang kamu faham tentang transcription? Okay? So, kalau kita tengok overview, the Francis Crick in 1956, they named the flow of information from DNA into RNA, into proteins, the central dogma. Because DNA can go to RNA and then protein can, but the protein cannot go to DNA, cannot go to RNA, cannot go to DNA. They cannot reverse. Yeah, and this is what I call the central dogma. And synthesis template is what we call DNA transcription. Synthesize the RNA molecule. What are the RNA molecule that they synthesize? Basically, is referred to the mRNA, or what we call the transcript, the sense strand, which is later be translated into protein. Okay, so this is the basic overview. So there are 4 major types of the RNA molecule during the transcriptions. Okay, first is the messenger RNA, right, which encodes the amino acid sequence of a polypeptide. Okay, this is the mRNA, the sense strand, the one that will be translated into proteins. The other one is a transfer RNA, what we call the tRNA. So bring the amino acid to the ribosome during the translations. Another one is the rRNA, which is a ribosomal RNA, combine the protein to form a rhizobome, and that's catalyzed for translation. Okay, the catalyst lah, satu stimulants, a catalyst for translation. And the other one is the small nuclear RNA, the snRNA, which combine with the protein to form a complex used in the eukaryotic RNA processing. Okay, RNA processing, also called RNA splicing. That's what we're going to talk about later. Okay. Now, the transcription is regulated by the gene regulatory elements associated with each gene. So you have the polymerase as well, start of the transcription, and then the direction. This is a direction, then you can see how they actually transcribe it. Okay, and make the RNA. So the DNA will be unwound. Okay, they are unwind in the region next to the gene due to the RNA polymerase in the prokaryotes or other proteins in the eukaryote. They want to synthesize the protein. That's why they need to transcribe it into the mRNA first. So the RNA polymerase will catalyze the transcriptions, and RNA transcribe is from a 5' to 3'. Okay, and the template of DNA's strand is read from 3' to 5'. So it's a complementary DNA, and the non-template strand has the same polarity as the RNA. So RNA polymerization is similar to the DNA synthesis, except first the precursor are NTPs, not DNTPs. All right. If you look at the RNA polymerizations and DNA polymerizations, the differences, okay, because the other one is the oxy, the oxy. This one is the oxy. Okay, so they only use NTPs, not the NTPs, and no primer is needed to initiate the synthesis for RNA polymerization. For DNA polymerization, yes, they need proteins, and the uracil will be inserted instead of the thymine. So the U will be replaced, the T will be replaced by uracil, not the thymine. Okay, boleh terima?
Boleh.
Boleh. Yang inilah, bagaimana the growing of the RNA strand. Kalau kita tengok, so they read from 5' to 3', but the DNA template is from 3' to 5' kan? Ya, di sini. Ya, this is the template 3 to 5, but the one that growing of the RNA strand is 5 to 3. Dia mesti 1,3, 1,5. So the RNA polymerization form the RNA. So you can see T again pair with A, and G they pair with C, but A They don't more pay with T, but they pay with
U.
U kan?
Yeah.
Yes.
That's the difference. Ya. Kalau DNA ini ialah T kan?
Yes.
Okay. Kalau RNA ini ialah U. So we call AUGC. Boleh ya? You see the differences between RNA polymerization and DNA polymerization? Can you see the difference now? Okay, very good. So the transcriptions is divided into 3 steps for both prokaryote and eukaryote. First, of course, they need to initiate. After the initiations, they need to go for elongation. And after the elongation, they read and read and read and read, then they need to terminate, they need to stop. So, boleh faham ah? So, dia macam ayat. Okay, IET, ayat. Initiation, elongation, and termination. 3 proses saja. Boleh ingat tak ni? Dia mesti initiate dan dia mesti start kan? They need to start. Then after the start, they need to what? They need to do reading. Reading is elongate lah. Dia memanjangkan to get the RNA strand keluar kan? Then after certain point, they have to be stopped. Dia mesti terminate. So it's a termination. Okay? So elongation is highly conserved between the prokaryote and eukaryote, but the initiations and terminations are somewhat different. So in prokaryote, the gene involved in transcription basically has 3 regions. First, they have the promoter. Promoter ialah promote the process. Okay? Attract the RNA polymerase to begin the transcription at the site specified by the promoter. Yeah, they promote the transcription. Okay? And the transcription sequence, the transcript sequence called the RNA coding sequence, correspond with the RNA sequence of the transcript. So ini. Okay? Dia mesti ada di depan tu, dia mesti ada promoter. Lepas tu tengah-tengah ialah satu strand di mana yang dia kena transcribe kepada RNA itu. And the last one is the terminator lah. The downstream of the RNA coding sequence that will specify where the transcription will stop di sini pada akhirnya. Okay? Ada permulaan oleh promoter. Lepas tu kita ada RNA coding sequence di mana ini adalah gene. The gene will be later, kalau dia mRNA, it will be later be translated into protein, and after that they have the termination code dekat sini. Boleh faham ya?
Boleh.
Okay.
Boleh, doktor.
Okay. So the promoter in E. coli generally involve 2 DNA sequences centered at the 35 base pairs or 10 base pair upstream of the +1 start site of the transcription. Kita ada transcription start site. Okay, so 35 or 10 upstream of it. Itu ada region di mana kita boleh cari promoter. Boleh? Promoter dia adalah di upstream, di atas, di depan the transcription start site tu. Boleh?
Boleh.
So kalau kita nak, contohnya kita nak tahu fungsi gene ini, kita knock down the function, boleh tak? Kalau kita nak faham fungsi gene ini. Betul? Kalau kita nak tahu fungsi gene ini, kita boleh tak buang dia punya, kita boleh knock dia down tak? Boleh knock down. Kalau knock down, supaya gene tu tidak di-express. Boleh tak? Boleh kan? Betul? So, salah satu daripada caranya ialah kita knock down the promoter region. Bila promoter tidak ada, dia tidak boleh promote the transcription start site kan? Dia tidak boleh promote transcription kan?
Yes.
Ya, bila transcription tidak berlaku, maka tidak ada produk protein tu, betul tak? Because the protein is the one that doing the function, betul tak?
Yes.
See, ini adalah antara satu caranya. Kita knock down the promotion region, promoter region tu. Bila sampai second year nanti, kita akan belajar tentang knock down, how to knock down animal punya function, knock down the protein or knock down the gene. This is one of the method lah that we can use. I will teach you in deontology. Memang saya seorang ajar deontology. So I'll teach you on that. Okay, so the common E. coli promoter that is used for most transcription has this sequence, which is first is the 35 regions of the consensus of TT-GACA. The reading is from 5' to 3', or the -10 region, the consensus sequence of the TATA, -5, the 5' to TATAAT, to that sequence, the short sequence. Biasanya kita boleh dapatkan promoter in E. coli. Okay, then that's the promoter region, the promoted. So the transcription initiation require the RNA polymerase enzyme to bind to the promoter DNA sequence of that consists of first the core RNA polymerase and the sigma factor. Where the sigma factor, they binds the core enzyme and confer the ability to recognize the promoter and initiate the RNA synthesis. So without the sigma factor, the core enzyme randomly bind to the DNA. So dia tidak bind to the promoter region. Yeah. This is the transcription in prokaryote. So the RNA polymerase bind promoter in 2 steps. First is bind to the -5 sequence, 1. Another one, second, is bind tightly to the -10 sequence, the positions to begin the transcription. So once the initiation is completed, the RNA synthesis will begin. Yeah. So after the 8 to 9, the NTPs have been joined in the growing RNA change, the sigma factor is then released and reused for other initiations. So the sigma factor can be reused. The core enzyme completes the transcription process. Macam di sini lah. Okay. Dia buka, lepas tu dia akan the DNA polymerase datang, dan dia terus akan do the elongation di sini, whereby they keep reading and synthesize the RNA strand. Boleh faham, kelas?
Boleh.
Boleh faham, ha? Kalau you ada baca before this, memang you boleh faham lah. Okay. So the core enzyme, they untwist the DNA helix locally and allowing a small region to denature. Dia mesti allow it to denature supaya dia boleh open up, because the DNA strand actually dia double stranded yang very tightly bound together kan. So dia mesti open up. So the newly synthesized RNA form a RNA-DNA hybrid, but most of the transcript is displaced as the DNA helix reform. So the chains grow at 30 to 50 nucleotides per second. Okay, and the RNA polymerase has 2 types of proofreading. First, similar to RNA polymerase editing, newly inserted nucleotide is removed by the reversing synthesis, the synthesis reaction. And second is the enzyme move back 1 or more nucleotide and cleave the RNA, then resume the synthesis in forward direction. Another one is the terminator sequence are used to end the transcription. There are 2 types in prokaryotes, what you call the raw independent and row dependent. Row independent, or the type 1, what we call the type 1 terminator. Row dependent is what we call the type 2 terminator. They must be terminated. Kalau tidak, dia akan keep reading. It's a waste actually, because gene bila dia sudah fully read by the polymerase, so it will bila sampai ke hujung, because setiap gene dia ada size tertentu, betul tak?
Yeah.
Ya.
Yes.
Kalau sudah the full size of the gene sudah reading, dia kena stop lah kan. Kalau dia tak stop, apa guna dia keep reading kan? Yeah, correct?
Yeah.
So dia stop. So complete lah. Contohnya mRNA itu kalau sudah complete, and after that they'll go for the RNA processing, di mana intron tu akan remove nanti. Okay. So transcription in prokaryote terminations does not need raw protein to stop the transcription. So in this mechanism, transcription is terminated due to the specific sequence of DNA terminator, the terminator in the terminator DNA. So the terminator DNA contain invert repeat, which cause complementary pairing as a RNA form the hairpin structure. Bila dia macam satu hairpin tu, so they will stop. So the invert repeats followed by the larger number of TTTTT, which is, for example, base first BP on the template DNA. So the uracil appear to be the RNA, and the lot of the hairpin structure is not tolerated by the AU. So the base pair, so the RNA gets separated from the RNA DNA heteroduplex. Right? So dia akan inilah this is the example. Dia A dengan U kenapa kan? Because U is in the RNA kan. So bila dia sampai sini, then this is what we call dia automated, dia akan terminated lah. Ya, because dia akan form satu hairpin. Tahu hairpin loop tak? Satu loop, tahu tak?
Yes.
Okay.
Yes.
Bila dia sampai ke ada allow a hairpin loop to form, maka that's the time that it will be terminated of the elongation process. So far okay tak?
Okay.
Boleh ya? Boleh ya? Okay. Tommy, boleh faham tak Tommy? Sudah tidur Tommy?
Okay, sir.
Okay ya? Okay, good. So the transcription in prokaryote, so the role dependent or type 2 terminator that require the protein of rho for the termination. So kita ada rho protein di sini. Okay, di sini, and then the terminate. So the terminator is a sequence rich in the C rich and poor in the G poor. So terminator does not form a hairpin loop di sini. Kalau type 2 lah. So the rho protein bind to the C rich upstream of the termination site, and the rho, which is the helicase, move the locations of the RNA polymerases. So the rho unwind the DNA and RNA hybrid, and then the RNA transcript will be released, and the RNA polymerase and rho will be disassociated. Akan dikeluarkan, maka dia akan berhentilah. Right? Kita ada dua termination kan. One is the rho independent, which is a type 1, and rho dependent, which is a type 2. And type 1 and type 2 terminator process, they are slightly different. Okay? Satu, type 2, type 1 tu dia gunakan A kan. Lepas tu dia akan form satu hairpin loop kan, and cause the termination. Yang di sini dia gunakan the rho protein. The rho protein akan dissociate dengan RNA polymerase. Okay tak? Can you see the difference of type 1 terminator and type 2 terminator?
Yes.
Yeah.
Can you see the difference?
Yes, doctor.
Ya kan? Okay. So kita berhenti di sini hari ini. So far okay tak? 1 jam setengah.
Okay.
Any question?
Okay.
Any question, class?
Doctor, about the terminator, we have the two types. Do we have a certain situation that we must use the type 1 terminator, or certain situation that must use the type 2?
It depends, I think. It depends on the what we call the, it probably depends on the environment that needed. Okay, or what they, probably when they are in the lacking of certain nutrients, so they are using type 1. If the other one, they probably use a type 2. It very much depend on the environment of this for the eukaryote. Sorry, for the prokaryote transcription.
Okay.
Okay.
Okay. Thank you, doctor.
Okay.
Oh, I have one question, doctor.
Yeah.
For our midterm question, can we review it or?
No, cannot review because it's all copyright. Kenapa?
Oh no,
You're
I
not
just
happy with the result?
No revision, doctor.
No, ini
Yeah.
university sudah lah, Amoy. Hey, come on. You are very lucky because I gave you MCQ. In university, I tell you what, in the university that I attended in Europe, in UK, we never have MCQ, you know. Even we have MCQ, it's very different than the MCQ that we have. If you get correct, they give you 1 mark. If you get it wrong, they minus 1 mark from you. Don't too worry about this question. You need to worry whether you truly understand this subject or not. That is something that I really concerned about. Okay? Boleh tak?
Thank you, Doctor.
Jangan risau pasal
Boleh.
soalan. You bukan lagi macam sekolah menengah. Your teacher asks you to memorize. Then you read the exam, you just throw out, you vomit, and you get the marks, right? But do you really understand the subject? That matters, you know. In university, it's not the matter you're scoring. It's the matter that you truly understand and you can use the knowledge that you learned to apply to the work later, to solve the problem. That is something that we are looking for, not the marks anymore. Okay? Boleh,
Boleh.
Boleh, Doctor.
Okay? So I see you next week. I'm going to see you next week.